The first speaker is Jodi McGraw, who she and I have been worked together or known each other for 30 years that we've both worked in the Santa Cruz area. She originally did her undergraduate degree at UCSC studying conservation of rare plants in the Santa Cruz sandhills, then did her PhD at Berkeley, continuing that work, and for the last 30 years has a small consulting firm that specializes really on conservation of some of the rare plant communities in the area, and she's very kindly taken my class on a number of field trips out to see her work. And she is really known as the expert on the Santa Cruz sandhills, which are a really cool endemic plant community that she's going to talk about today. So I will pass it over.
So great. So as many of you guys know in the audience, various types of anthropogenic drivers are causing rare plant extirpation. So we're losing populations of our rare plants and our broader biodiversity, of course. And in some cases, introductions are going to be needed to ensure species persistence. And to be successful, these introductions must address various factors that are going to influence long-term persistence of populations. And unfortunately, a lot of these factors are oftentimes poorly understood. Experimental approaches can be used in introductions to fill these critical data gaps and ensure and promote long-term success of these introductions, which have notoriously not been very successful, as we all know from the literature. Let's see, I've listed here in the slide on the right some of the important introduction considerations, abiotic and biotic habitat factors, of course, disturbance regimes, and then important population factors like mating systems, genetic considerations are all part of the game. As Karen mentioned, I've been doing work in the Santa Cruz sandhills where we explored these concepts as for the last 30 years. And the Santa Cruz sandhills, if you're not familiar with them, are unique communities of plants and animals that are found only on outcroppings of sand soil in central Santa Cruz County. And they're home to 4 endemic plants and 2 endemic insects that have been described. And these are just the tip of the iceberg. There's many undescribed taxa. And the sandhills... Yeah.
Sorry to interrupt. Can you turn your camera on? That's why we can't see you.
So as I was saying, the sandhills have a really important disturbance ecology, which is an important part of the successful introduction. They're fire-adapted systems, so fire at a large spatial scale, maintains open habitat conditions, removes shrubs and trees. At smaller spatial scales, though, there's a lot of soil disturbances on the sandy system. The slides, erosional features, trails, and gopher mounds all play an important role in maintaining open conditions. They remove accumulated leaf litter and thatch on the soil surface. They reduce competition, particularly from herbaceous exotic plants, and they can stimulate seed germination in some cases. The sandhills have been lost, threatened by a lot of factors. There was only about 6,300 acres originally. We're down to about half of that. Much of that acreage I'm listing here has also been degraded by development. And the remaining intact habitat has been impacted by fire exclusion, which has caused unnatural succession, exotic plants, which have invaded the system, and incompatible recreation use. And then climate change in the last 20 years has created this horrible overlay of factors that has increased the climatic water deficit. We have these mega droughts, we have these increased temperatures, all of which are exacerbating the naturally droughty conditions and causing declines in the populations. And so Ben Lomond Wallflower has been the poster child for these impacts. This species, Erysimum teretifolium and the Brassicaceae, is a biennial to short-lived perennial herb. It's generally monocarpic, which means it reproduces just once. It's endemic to the sandhills. It's been listed as endangered for more than 40 years. It has a weakly self-incompatible mating system, which means there is some self-fertilization that occurs, but there's also late-acting inbreeding depression where closely related individuals have lower survivorship and reproduction. The populations preferentially occur in open canopy habitat away from shrubs and trees that's normally been maintained by fire. They also require persistent preferentially in these open soil conditions created by slides like this shown here. They're strongly outcompeted by herbaceous exotic plants that are ubiquitous, unfortunately. Many seedlings, about 95% by my prior count, have died before they even reach the juvenile stage, much less try to reproduce. As a result, about 5 of the 17 populations that were documented in the 1970s have been extirpated. One of those populations was at the Botany Ecological Reserve, which is shown here. It's one of the disjunct populations of wallflower, and it's about 552 acres is the entire preserve. It's really was protected from being used as a vineyard back in 1989 by the Nature Conservancy. It's now owned and managed by the California Department of Fish and Wildlife. And it had two populations up until the early 90s. The northern one has been extirpated about in 1994. The southern one has about 9 individuals as of the count this year. So I and my collaborators wanted to conduct an experimental reintroduction to get this population secure and expand it. Our study questions were, what are the effects of soil disturbance on wallflower? What are the genetic aspects of an introduction that should influence wallflower introductions? What habitat conditions are most important to consider? We also had a series of project goals examining the alternative soil disturbance treatments that could be done to promote the species, evaluating whether Bonny Doon population features locally adapted genetic complexes that should be considered in future introductions, or alternatively, whether we want to alleviate the inbreeding depression that's being caused by reproduction amongst closely related individuals as the populations get smaller. We also wanted to determine suitable habitat conditions for the populations, and then finally just establish wallflower in multiple locations. So we used a small-scale manipulative experiment. It was replicated in 2 years. So we essentially did the same experiment in 2 separate years because we know that interannual variability and weather can have strong influences on the success of this introduction and our findings. The 1 year, 2019, was a really high rainfall year, 180% of average rainfall, and 2020 was a drought year. So it's good that we did that. We monitored each introduction for 2 years. We replicated it in 2 different community types, the northern maritime chaparral, as well as maritime coast range ponderosa pine forest. We had 2 disturbance treatments, rake only, where we removed all the thatch on the soil surface and all the established plants using rock rakes. And then we also had a tilling treatment where we, after raking, then turned all the soil over using shovels to a depth of about 30 centimeters. Very labor intensive, not something you would do full scale. We're looking at methods to scale that up, of course, but we wanted to create the loose sandy soil conditions that wallflower has been found to be associated with. Our experimental block design shown here. Essentially, we had 2 plots of each of the 2 disturbance treatments, plus another plot that we didn't seed, but we did submitted to the that we subjected to the same treatments to look to see if there was a seed bank. There was no seed bank. Wallflower did not come up in those plots, so I won't talk about those further. This is an image showing our plot design. There are 2 meter by 2 meter plots. We had an interior seed plot that we seeded with a very large density of wallflower seeds that were collected from the Bonny Doon population. And then around the seed plot, we had 36 outplanted seedlings that were created in a greenhouse by careful crossing of wallflower collected from different populations. We have a non-Bonny Doon population crossed with another non-Bonny Doon population to look at heterosis amongst those 2 populations. We had a single non-Bonny Doon other population. We had Bonny Doon crossed with non-Bonny Doon, and of course, just the Bonny Doon genotype by itself. Let's see, we had data collection. In the 2 years, we collected data on soil conditions, soil depth, texture, chemistry, also plant community composition. And we looked at the effects of this experiment on 2 other rare species that also occur in the system, Ben Lomond spineflower and Ben Lomond buckwheat. For wallflower, we took a demographic approach. So we followed individual plants with toothpicks to look at their establishment, survivorship, reproduction, which is the proportion of plants that reproduce, and also the seed production, which is obviously how much seed is produced, excuse me. So what did we find? In these graphs here, the top row will be year 1, the bottom row be year 2, rake is gray, till is green, is what we're comparing. And as you can see, there was no effect on establishment in either year, but the tilling treatment increased wallflower survivorship in year 1. Tilling also promoted the reproduction of wallflower, so a higher proportion of the plants that survived actually flowered. And then, of course, they also had higher seed production, a higher number of seeds were produced. Looking at the transplanted seedlings, rake again is on the left, till is on the right. We found that tilling promoted the survivorship of plants in year 2 down here. The seed production was benefited in both years. But seed production, the actual number of seeds produced, was not increased by the tilling treatment. So overall, we have a benefit of tilling. There were no benefits of raking. What's the mechanism behind that effect? We're still scratching our heads a little bit on this. It's definitely an effect of reduced competition, of course. We did not detect any effects on soil nutrients. There was no increased nutrient flush or anything like that that we detected anyway. But there's something about the loose soil that seems to benefit wallflower. So here's kind of what that looks like. You can see that right out of the gate, the rake plants are smaller, and across the 2 years, it just kind of that gap just widens. You can also see that plant cover on the raked plots is higher than the till plots. So some of that is definitely a competition story. And we can see that wallflower size is negatively correlated with exotic plant cover at the block level and also with native plant cover. So co-occurring plants, native plants are also competitors for wallflower at the plot level. What about the genetic treatments? What did we learn from that? We found that actually the different genotypes, their performance varied by year and by life stage. So we didn't have any consistent evidence for either local adaptation, which is what we'd expect if the Bonny Doon genotype did better, or inbreeding depression, where the crosses did better, or outbreeding depression, where crosses did worse. So we did find that the hybrid genotype, the Bonny Doon crossed with a non-Bonny Doon, had higher performance in 5 out of the 7 metrics that we looked at, including survivorship. So this does have some evidence for perhaps for heterosis, which is where non-closely related individuals do perform better when they reproduce. What about habitat conditions? We did analysis of use non-metric multidimensional scaling to look at plant community composition and use PCA to look at soil conditions. And we found, in short, that wallflower performed better in the lower fertility soils, which had lower exotic plant cover. So it's an indirect effect that the higher fertility promotes exotics, which negatively impacts for wallflower. So PCA factor 2 is an indicator of negative soil, lack of soil fertility, and you can see wallflower size and survivorship were both positively correlated with this lower fertility soil. We were able to get wallflower established in all 8 of our block locations. So these are the 8 locations, and these are all the different metrics. This is a little color metric scale showing from brown, not so good, to green, good, how wallflower did in the different blocks. And you can see what we found is that the performance of wallflower differed among the blocks in different measures. So for example, and they weren't correlated. So in one of the chaparral blocks had really high survivorship. 93% of the seeds produced a plant, which is amazing, but then had lower performance of survivorship and reproduction, whereas one of the parkland blocks had much lower establishment, much higher survivorship and reproduction. So that's kind of just interesting that habitat is interacting with different life stages to influence how things work out for wallflower. So what did we learn and what are we going to do? The implications for our introduction are that we definitely think a disturbance treatment is going to be needed, and tilling is or scaling, loosening the soil through some mechanism that simulates what tilling does is going to be really beneficial. We're also interested in looking at how low intensity, low frequency soil disturbances that are ongoing can help maintain those loose sand soil conditions. In terms of genetic considerations, genetic mixing is unlikely to disrupt local adaptation. So there doesn't seem to be that the Bonny Doon genotype is important to long-term persistence there. And instead, it suggests that maybe alleviating self-incompatibility, which is resulting from the really small population, is going to be our best bet. And then in terms of habitat, we believe that reintroducing in a variety of habitat conditions is going to be most beneficial long-term to kind of capture those, again, interacting influences of interannual variability in climate with habitat conditions, hot sites, dry sites, moist sites, et cetera. And also the overall just having areas that have reduced exotic plant competition or managing exotic plants is going to be really important. We learned some other important things I thought I'd pass along. Outplanting seedlings can really jumpstart the introduction. It's kind of not typical to outplant seedlings if you have like a biennial perhaps, but our transplants flowered at really high rates in just their first year as a result of being grown initially in the greenhouse. And they also had much higher seed production. So it's kind of like a little head start program for a rare plant introduction to outplant those. Having larger treatment areas is something that we know will be really beneficial. We had some edge effects from herbivores chewing up our plants, and also the exotic plants kind of re-infiltrated our plots pretty quickly following the treatments, but having larger treatment areas will alleviate that. And we were encouraged to see that Ben Lomond spineflower and Ben Lomond buckwheat also recruited at really high rates in response to the treatments, which also promoted a larger assemblage of native herbaceous plants to the system. So currently we have wallflower persisting in all 8 of the blocks. So that's pretty exciting because we had like a multi-year drought since we established this introduction. We also had the CZU lightning complex fire burn part of our treatment areas and some of our blocks. And also there was a bulldozer line that went through some of them. We've also had to borrow some of the seed for our next phase, which I'll talk about briefly. And then we have also seen wallflower dispersing outside of the treatment plots into the adjacent habitat, which of course is the real goal for this introduction. So we have a current project, which we're just wrapping up, which we're using that CZU lightning complex fire as a natural experiment to see if we can leverage it and learn more, as well as establish wallflower further. So we seeded wallflower in the areas that were burned, as well as areas created by the fuel break, which is kind of like was done with the dozer, kind of like a larger scale version of our tilling treatment, we hope. And so we have some larger scale plots, and we're looking to put trails through those plots in order to maintain loose soil conditions over time. So with that, sorry for the slow start there in the tech glitch, but I'd like to acknowledge all the contributions of all of our collaborators on this project and our funder, the California Department of Fish and Wildlife and the U.S. Fish and Wildlife Service. And I'm happy to answer any questions if there's any time. Thanks.
Okay. Thanks, Jodi, for fitting a lot of information into the time allotted. And so maybe we have time for 2 quick questions, I think. I guess I will call on people since she can't see you. Anybody have any questions? I'm looking. Oh, yeah, go ahead.
Well, here, I'm bringing the mic.
Thank you. Are you considering any genetic sequencing to determine population variability?
Did you hear that, Jodi?
Yeah, I could. Yeah, that work has been done by my collaborator, Justin Whittall. So he's got some papers out looking at the actual populations, and I didn't have time to talk about that. So yes.
Okay, and do you have, and then maybe one more question? There's somebody else? Yes, right there. Yeah, pass the microphone. What? Are we supposed to go to the back? Okay, we'll go to the back there. Go ahead. Yep.
I worked on some Enotheras and Camasonias and found that the gopher burrow sites were really hot for sexually reproducing adults, and where I had self-feeding populations, they did very poorly out in the US. But wherever there was gopher mounds, I was able to see sexually outcrossing individuals appear, which was really cool. And I thought you said that you had positive effects of gopher burrowing. But the gophers are not grazing the plants, right? Or are they?
No, they do that too. But I do think that the net effect of gophers is positive. The soil disturbance tilling effect, I mean, I haven't actually quantified that, but I do believe in general that gophers, by tilling and maintaining loose soil conditions, have a positive effect on wallflower above and beyond what their herbivory. They do take wallflower.
I just wonder if you could in parallel have somebody study the activity areas of gophers and see if you can improve the gopher activity to increase the wallflower as well.
We don't need to improve the gophers. They're doing a good job of their own out there, as probably everywhere else. So good idea.
I think Jodi's answered them so quickly. We'll take your one last question up here.
I'm curious about the pollinators out there. Are there pollinators visiting these that are also using plants around these small populations?
Yeah, I would refer you to Miranda Mellon's work on that. She has studied the pollinators of wallflower, and some other folks have as well. There's a Chalcedon checker spot butterfly, which is a generalist pollinator. Bombus. They're basically not. They don't have any specific pollinators. I've understood from some other studies that have been done that they are like an important part of the pollinator network in the sandhills, which is super exciting. But there's butterflies, bumblebees, and I believe many other species that do pollinate these.
Okay, thank you, Jodi.
Thanks.